Okay, in the fifth and last part of this lecture, we'll discuss about the blood. So, the blood consists of the plasma and its formed elements. So, basically, it's not a solution, it's more of a suspension, or no, not a colloid, it's a suspension. Blood is critical in maintaining homeostasis. It regulates the internal environment of the body by diffusing ions and other materials into the interstitial fluid. It forms clots to prevent blood loss at injuries. It transports heat, hormones, nutrients, gases, and ions. It also provides protection through the leukocytes and antibodies. Actually, it works in relation to, of course, your immune system and the lymphatic system. So if you look at our blood, it's, as I said, it's a suspension. So you have 46 to 63% of plasma, which is basically the liquid part. This 92% water and dissolved plasma proteins and other solutes, so 7 and 1%. And then we have the formed elements. The formed elements are the erythrocytes, also known as the red blood cells, the leukocytes, which is the umbrella term for the different types of immune cells, the white blood cells, and then the thrombocytes, we have the platelets. So this is hematopoiesis, or basically the formation of blood cells. So it started with a stem cell. A stem cell is a cell that has no specialization, so it can differentiate into specialized cells. So in the bone marrow, you have stem cells forming a megakaryocyte, which forms your platelets. And then you can have a reticulocyte, which rejects its nucleus. So by the way, unlike other eukaryotic cells, your red blood cell do not have a nucleus. Okay, so it was ejected after forming the erythrocyte from the reticulocyte. Now, the bone marrow cells, the bone marrow stem cell, also known as the myeloid cell, can also form some of the known lymphocytes, or the white blood cells. So we have the eosinophils, the basophils, the neutrophils, and the monocytes. Monocytes are macrophages, basically, within the bloodstream. So you have your, these are, each of them have their own task in defending the body against invading pathogens. So aside from the bone marrow, we also have lymphoid stem cells that forms the T and the B cells. The one of the more better known white blood cells. Actually, between characterization of these blood cells, I'd refer to you to the anime Cells at Work. It's actually quite a very good anime and explains a lot about the blood cells, and of course the immune system. Now let's look at the white blood cells. The white blood cells, or leukocytes, these are defensive cells. They are very critical to the immune system. We have 5 types of white blood cells. We have granulocytes, which have a lobe nucleus and visible granules. So this includes neutrophils, eosinophils, and basophils. We have agranulocytes, which have spherical nucleus and non-visible granules, the lymphocytes and the monocytes. And you have neutrophils, which are the most numerous, and the basophils. Among them, neutrophils are the most numerous, and the basophils are the least numerous. So you have basically, never let monkey eat bananas are just the mnemonics for the different white blood cells. So never neutrophil, let for lymphocyte, you have M for monocyte, E for eosinophil, and B for the basophils. Okay, so basically you have those. Okay, now how about the red blood cells? The red blood cells, or the erythrocytes, they transport oxygen to the tissues. They are the most numerous blood cell. So each red blood cell carries approximately 200 million hemoglobin molecules. And as we discussed in the previous lecture on the respiratory system, each hemoglobin can carry 4 oxygen molecules. So the heme group in the hemoglobin binds your iron, and which picks up the oxygen in the lungs. Okay, so the RBCs circulate approximately 120 days before they are damaged enough for removal in the circulatory system. So you're changing your blood for every 120 days. So the spleen and the liver remove worn out red blood cells, breaking them down and recycling their constituent minerals and proteins. So we have around 2 million RBCs broken down per second, and we produce RBCs at a rate of 2 million cells per second. So the rate of RBC formation is affected by the hormones and environmental need. So hypoxia, or low oxygen when we encounter hypoxia, basically the body's oxygen level is lowered. So that is the stimulus for erythropoiesis, because it means that the oxygen in the body is not distributed well enough. So we need more work or something like that. So red blood cell surface proteins also determine blood type. So we have the four blood types of the ABO blood system: A, B, AB, and O. So these are identified by protein markers or the antigens. Although antigen is more of a generalized term, but antigen is basically just the molecules where antibodies attached to. Okay, so anyway, these are protein markers in the surface of the red blood cells. So the O blood type lacks these antigenic markers or these protein markers. So anti-agglutinins or antibodies. So in the blood plasma, we have anti-agglutinins and antibodies. So sorry. So basically, these antibodies distinguishes the self from the non-self. So if you are a non-self, automatically the antibodies will mark you for attack by the white blood cells. So it's also important in blood transfusion, because if you have blood type A blood, you have an anti-B antibody in your plasma. So a cell with blood type B or with a B antigen cannot enter your system. So that means you cannot have blood type AB or blood type B transfused to you. So you can only accept blood type A having the same antigen as yourself, or O, whereas it has no antigen whatsoever. So it can be you're actually safe from any attack of your anti-B antibodies. Whereas the same goes for blood type B. So blood type B cannot have, since it has anti-A antibody, so it cannot receive any blood that has a marker A. So that means no blood type A nor blood type AB. It can only accept blood type B and blood type O. So this is why blood type O is called the universal donor, not because there are many of blood type O's, but rather blood type O has no antigen. So the anti-A, either anti-A or anti-B antibodies in the plasma of the blood will not react with blood type O because it has no antigen whatsoever. On the other side of the spectrum, you have AB. AB blood is the universal acceptor because they do not have anti-A nor anti-B antibodies, because they have A and B, both A and B antigens. So they can accept almost any blood type. Although that doesn't mean that you can just transfuse any type of blood. So there, AB, and O are just some of the blood types. Since then, there have been many other different blood types that have been discovered. Okay, so these are basically your antigens. So antigens present in the surface of your red blood cells. So you have type A, type B, and in the type AB, both antigens are present. In type O, neither are present. So if you have your the issue here is your plasma agglutinins and your plasma antibodies must not attack the receiving or the incoming or the donor red blood cells. Otherwise, it will be bad for you. And aside from the ABO blood system, you have the Rh factor, which is determined by antigen D. So the presence of antigen D makes the blood positive. So the issue with Rh factor is apparent for pregnant women. So if we have Rh negative mothers, that means they do not have the antigen D. They of course develop, although they are not always having that, but they will develop when they carry Rh positive fetus. They will develop antibodies for Rh, for the antigen D. So what happens? Since antibodies can pass through the placenta, the antibodies can attack the blood of the fetus, and so it can be fatal to the fetus. So we have methods to prevent that by injecting them with ROGAM shot, which can destroy the Rh positive blood and prevents her body from making anti-D antibodies. So ROGAM is basically it's a molecule. It doesn't mean that it will kill the fetus or the blood in the fetus, but any Rh positive blood that enters the woman's body. Okay, so aside from the red blood cells, we also have the third formed bodies in the form components of the blood, which are the platelets. They are fragments of large cells called the megakaryocytes. So they contain proteins and acids. They're actually quite one of the smallest cells in the body. So when released, they began a series of events that leads to the formation of a blood clot. So hemostasis is the process of repairing a hole in the blood vessel through the clotting cascade. So it starts with a vascular spasm to reduce blood flow and blood loss. Then the platelet flag will form to temporarily seal the wound. And a true clot forms through intrinsic and extrinsic mechanisms. So basically, it's actually quite, the pathway here is more of a cascade of a biochemical reaction. So we won't dwell too much on that, but just that at the end, you will form basically a netted webbing of basically the platelets forming fibrin threads. Okay, so these are from the platelets. So they hold the red blood cells so that they won't basically exit the body through the break in the wall, in the blood vessel wall. And then we have the different blood disorders. So infectious mononucleosis is a common blood disease on college campus and is caused by an Epstein-Barr virus and transmitted through saliva. So carbon monoxide binds to hemoglobin in place of oxygen and leads to hypoxia, followed by disability or death if left untreated. So carbon monoxide binds to hemoglobin 200 times. I think I discussed this one last time during the discussion of the respiratory system. Carbon monoxide binds strongly to hemoglobin, more strongly than oxygen. So the Epstein-Barr virus can live in the can be basically one of the basically infectious diseases that can affect your blood. Now, other pathogens can live in the blood. Hepatitis, HIV, and other sexually transmitted diseases are found in the blood. It is important to be careful when dealing with and handling blood to prevent infection. So basically, blood is one of the most effective transmitting agent. I believe it's Ebola. The outbreak of Ebola exposure to the blood of Ebola patients can infect you with Ebola. Donated blood is screened for pathogens to prevent transfusion of tainted blood. So other blood disorders include leukemia. Leukemia affects white blood cells and includes several cancers of the bone marrow. So in most leukemias, the white blood cells are shaped abnormally and do not function properly. So symptoms are flu-like and include fatigue, weakness, compromised immune defense, swelling of the lymph nodes, and the spleen. So they are classified by its pattern of onset or by the specific cells affected. We have acute leukemia, which appears quickly. It fills the blood with immature white blood cells called blasts. Whereas chronic leukemia, it's more slow. Cells are more developed but still immature. Other blood disorders include anemia. Anemia is the reduction in the red blood cell population and thus in the blood's oxygen carrying capacity. So its symptoms include fatigue, weakness, shortness of breath, and sometimes chest pains like angina. So anemia is classified by the cause of the red blood cell deficiency. So it can be iron deficiency anemia. So you have shortage of iron leading to fewer hemoglobin molecules for each RBC, or even pernicious anemia, shortage of vitamin B12, which also inhibits RBC formation. Aplastic pneumonia, the bone marrow does not produce enough stem cells, resulting in this type of anemia. Hemorrhagic anemia, excessive bleeding that results in, of course, loss of RBC. And sickle cell anemia. Sickle cell anemia is a genetic disorder. So you have, the problem here is not in the RBC but rather in the hemoglobin. So it misshapes because of the defective hemoglobin. It changes the shape of the red blood cell. So instead of the cute donut shape red blood cell, you have a sickle elongated red blood cell. This causes an issue with, of course, first of all, hemoglobin cannot carry oxygen properly. It can only carry one oxygen molecule instead of 4 for sickle cell anemia. Aside from that, because of the misshapen red blood cells, there is a large tendency for blood clot to form because if you remember, capillaries are very, very small blood vessels. Sometimes they are very small, such that only a single RBC can pass through. Because of the donut circular shape of the RBC, there are no corners in your RBC, so they won't clog. But if in sickle cell anemia, they are elongated, they have corners, they can tend to basically clog one another in those very, very small blood vessels. But another upside in sickle cell anemia is that they're less susceptible to malaria and dengue. Okay? So anyway, that's it for the cardiovascular system. See you in the next lecture.